Welcome everybody to today's evening at Whitney's lecture. I'm glad to see you all here in person at the Lohmann Auditorium. And I also welcome everybody at home at the Zoom link that took us a little longer today to establish. So my name is Sandra Lösken. I'm an associate professor of chemistry here at the Whitney Lab at the University of Florida. And I'm excited today to be your host. As you all know, but people not many online might not know, that the Whitney Laboratory was funded in 1974 and is a biomedical research institute of the University of Florida. It's composed of currently 11 faculty, and we're actively recruiting a new faculty colleague. And we're using, as you know, marine organisms for biomedical research that can apply to human health, understanding of natural resources and ecosystems, as well as enabling to understand better basic biology, genomics, and evolution. So the Whitney Laboratory trains many scientists of all levels. We have from kindergarten to college level students here. We offer these monthly lectures for our community that you all take advantage of. And we also offer other programs. Recently, we added a lifelong learning program that is online available. So please check it out on our Whitney website. So definitely lots of educational programs available. We have the Sea Turtle Hospital that promotes ocean awareness and is part of our conservation through sea turtle rehabilitation, research, and education. So as you know, the Whitney Laboratory welcomes you to the new year, the Year of the Dragon, so it's a strong one. And I'm today very excited to host a good friend and colleague, Dr. Christine Salomon. She came to us from the University of Minnesota. As you can imagine, it's a nice change in temperature coming here to Florida. So Dr. Christine Salomon is Associate Professor at the Center for Drug Design at the University of Minnesota. She is a fellow chemist, and she's focused on finding new drugs from natural resources. Her work is funded by federal agencies and recently by the National Institutes of Health. Her work has led her into exploring caves, mushrooms, and lots of infectious diseases that she will share today. When I met her a few minutes earlier today, she also disclosed she is an avid ice dipper, I think is a word. And she's been a part of medical studies to look for the benefits of these ice dipping. So maybe there's something we can find in Florida as well. So without further ado, I'm very excited to have Christine Salomon here visiting us. Thank you.
Thank you. Well, good evening, everyone. Thank you so much for spending your evening with us here. Thank you for the kind invitation. This is the very best weekend to be out of Minnesota. I learned that on Monday, I think with wind chill, it's gonna be minus 20. Fahrenheit, yes, Fahrenheit. At minus 40, Fahrenheit and Celsius are the same. That's when schools close in Minnesota. So today I'm going to talk to you about one of our projects focused on a fungal disease of bats called white nose syndrome. And I'm really happy to see all of you here because this is not a disease that is currently in Florida, but it is affecting the whole rest of the country as well as Canada. And so I'm excited to tell you a little bit about the work that we're doing on this project, some of the progress that we've made, some of the challenges that we've run into, and some of the additional questions that we've developed along the way. But before I do that, I want to tell you a little bit about what I do in my laboratory. So like Sandra, we do natural products chemistry discovery. Mostly we work on trying to find new biological control agents. I'm going to tell you what that means, but we're looking for living things from nature that can fight things that we don't want to happen. So things that could fight pathogens or things that could fight pests that are infecting plants, for example. We also work on a lot of human diseases. So we're very interested in finding natural products to treat infectious disease in humans. So primarily antifungals, but we also look for antibiotics and antiparasitics. In all of this work, we're looking from nature. So we're interested in looking for new microbes. And I'm going to tell you about how we're looking for new microbes in underground locations, from subterranean locations, and why this is such an amazing resource for us. And also, through all this work, we're also using a lens of chemical ecology. So one way that you can do natural products discovery is to just go out into an environment, collect lots of different things, fungi, mushrooms, sponges, all kinds of things, and then just look for new chemical compounds. And we definitely do that. but we also think about why and where compounds, biological compounds, might be present in the environment, where species might be interacting and fighting each other, where they might be trying to protect resources, or where they might be trying to inhibit something else. So we use this lens of chemical ecology in all of our work. And I know that many of you are not necessarily chemists, and I only have 3 slides with chemical compounds on them. So I promise, Sandra, this would not be a chemistry talk, but I want to explain to you the basis of the work that we're doing. So in my heart, I'm a chemist, and I study the chemistry of nature. And so I want to tell you what natural products are. So how many of you have had a cup of coffee, a cup of tea, smoked a cigarette, or taken an antibiotic in the past week? Everyone. So you have all benefited from secondary metabolites produced by nature. These are all compounds that are made naturally. Of course, caffeine and nicotine are made by plants. Penicillin is made by a number of different fungi. Secondary metabolites are just thought to be compounds that are beneficial to the organism as a whole. They're not involved in primary metabolism, but they're thought to be important to the organism. And they're made by all living things: bacteria, fungi, protists, plants, and animals. And to date, at least 200,000 unique natural products have been isolated and described and characterized. So with that background, I'm going to move into the body of my talk. And these are the things that I want to talk to you about today. I'm going to tell you about the subterranean environments that we do our field studies in. And then I'm going to introduce this disease called white nose syndrome in bats. I'm going to talk a little bit about chemistry. And then I'm going to focus on some of the challenges of applying what we've learned into the field. So way up on the top of Minnesota is our primary field site. This is the Pseudogymnoascus iron mine. This was the very first iron mine and the deepest iron mine that was built in Minnesota on the Iron Range. It was built in 1882, and it was active until 1962. It is a pretty deep mine. So when we go to the bottom of this mine, we get into it's like a cage. It's not quite an elevator. It's more like a cage. And there's a wire that goes into a wheelhouse, and there's literally a person sitting there with 2 levers, and they just call over on the radio and they just say, we want to go to level 27, and he just sends us down. So we can go down to the lowest level of the mine, which is about half a mile underground. Most of the mine looks like this. It's orange because it's an iron mine. The water that's coming into this mine from all different directions is very concentrated in metals, mostly iron. As this water reaches the surface in the mine, it's exposed to oxygen. So what happens when you get iron and oxygen together? You get rust. So it's a very rusty mine, but it's also very beautiful. This mine is like a playground for microbiologists. So there are at least 27 levels. Each level it's called a drift. It's a tunnel. Each tunnel is about 2 kilometers long in both directions coming out of the central shaft. And each of these drifts is different from every other one. So for example, one of the drifts has extremely high levels of copper in the water. That copper precipitates out onto the rocks just on that one level. Copper is very toxic in general to microbes, particularly to fungi. In fact, we incorporate it into paint on the bottom of boats to inhibit biofilms and other kinds of growth, barnacle growth on the bottoms of boats. It's very toxic. And yet we can sample these rocks and we can find life. We can find bacteria and fungi that have evolved to live in this pretty toxic environment. And there's all kinds of interesting bacteria and fungi that just grow in this subterranean environment. So we had been collecting microbes from the Pseudogymnoascus iron mine for several years. We were primarily looking for antifungals for humans. You probably know that there's a big problem with drug resistance. We really need new antibiotics for bacterial infections. The same is also true for fungal infections. So we had been working on these other projects related to humans for a few years, but then in like 2008, we learned about this disease called white nose syndrome in bats, and it had just been discovered in 2007. This is a brand new disease. It did not exist before. These bats were only found in upstate New York in a single cave in the winter of 2006 to 2007. And it was discovered that this disease of bats was caused by a fungus called Pseudogymnoascus destructans. Isn't that a great name? This is a really strange fungus. It's psychrophilic, which means it's cold loving. So this fungus cannot grow above 18 degrees centigrade, which is quite cold for a microbe. It causes incredibly high levels of mortality for bats that are susceptible to it. So for some bat species, like the little brown bat, tricolor bat, northern long-eared bat, up to 100% of a bat colony will be susceptible, will die if the colony becomes infected with P. destructans. And as I mentioned, it was just discovered in 2007, and ever since then, it has been rapidly spreading across the United States and into Canada. So P. destructans is a fungus that is a dermatophyte. And you might be familiar with dermatophytes or like yeast infections. So, you might get a superficial yeast infection on your skin that causes some itchiness. PD is also a dermatophyte, but it can cause mortality. So when PD infects the membranes of a wing of a bat that is susceptible, and you look at it under UV light, you can see these little bright dots, and that's where the fungus is living in the skin, and it causes a lot of inflammation in the skin. It's not exactly clear how the fungus kills the bats, but there's a few reasons that are probably all true at the same time. And one of the most important is that it seems to cause the bats to wake up early from hibernation. So typically, a bat will go into hibernation for about 5 months. And over those 5 months, the bat will wake up once a month. And when it goes into hibernation, its body becomes cold. It becomes the same temperature as its surroundings. So what this publication shows is that when a bat is asleep, it's about 8 degrees centigrade. But then when it wakes up, a healthy bat, it becomes close to 30 degrees. And that's normal. This is what a normal bat does. So it goes into hibernation, it's cold, it wakes up, it's warm, and it does this about 5 times. Bats are kind of living on the edge, right? They eat as much as they can, they build up fat stores, and then they use those fat stores to sustain them through the winter. And then they use them a little bit to wake up. When a bat becomes infected with P. destructans, it starts hibernation normally, like healthy bats. But then you can see that towards the end of hibernation, it wakes up more and more frequently. And it doesn't have enough fat stores to sustain that many times of waking up. And so they can die of starvation. They will also often leave the hibernacula at this stage. So if this is, let's say, January in northern Minnesota, and they decide to wake up and leave the cave, you could imagine that that's detrimental. So there are often ravens now sit outside the mine entrance and they wait for these bats to accidentally come out of the cave and then they eat them. The other thing that happens with this fungus, this dermatophyte, is that it can cause very high levels of inflammation, and this can lead to perforation of wing membranes. So I mentioned that it was first discovered in upstate New York. And what this map shows, I know it's a very busy map, but all I want to point out is that this is where it was first found in a single cave in 2007. And then these colors show each successive winter where it was suspected to be found and then where it was confirmed. And so you can see that there's been a clear spread across the United States. It's currently been confirmed in 40 states and 8 Canadian provinces. Not yet in Florida. So this level of mortality for many bat species is completely unprecedented. There's no other infectious disease that causes this high of a level of mortality in any animal. And just to put this in perspective, so it's hard to think about numbers, but if we just hone in on the state of New York, before white nose syndrome hit New York, there were estimated to be about 1.5 million little brown bats in the state. Within the first 6 years of white nose syndrome hitting those bats, they lost at least 90% of their populations. And so by 2012, they only counted about 150,000. And this is really hard to recover from. So bats are long-lived and they don't reproduce very quickly. So even if they do develop some resistance or we develop some kind of treatment, these bat colonies will not recover within our lifetimes. So we don't exactly know what the impacts are going to be. We're just starting to see what those impacts might be, but we can imagine what they might be. We know that bats do a lot of agricultural work, particularly in places like the Midwest, where we grow a lot of crops, a lot of wheat and corn and soybeans that are affected by pests. And they're particularly affected by pests that fly at night or in the evenings. And so, people often think about bats as being a good control for mosquitoes, like you might want to put a bat box in your backyard to control mosquitoes. But the insects that they really love are these big, juicy insects. And these are the most detrimental insects for agriculture. So you can imagine that if we lose these populations of bats, there's going to be an increased cost to produce food, a monetary cost. There's going to be a cost to the environment, because if we don't have bats to control these pests, what are farmers going to have to do? They're going to have to turn to pesticides, for sure. So that's definitely a cost in lots of different ways. And then we don't really know this for sure, but because there are fewer bats, there might be more mosquitoes in certain areas. We might have increased risk of mosquito vector diseases. So you might be wondering, well, what are we doing about it? And lots of people are working on it, but there are currently no treatments for this disease that are used. There are lots of possibilities that a number of research groups are working on. So at the very beginning, I think the most promising thing people worked on was looking for like volatile antifungals. Like if you could just go in and spray a volatile antifungal into a cave, you could just kill this fungus. But if you think through that process, the logistics of that are so complicated. I'm going to show you some pictures of caves and you will see why it's just really tricky to do that. One interesting biological feature that was discovered is that P. destructans is very sensitive to UV light. So sunlight and UV light. And so you can use a strong UV light and you can kill PD in a matter less than a second, which seems really promising, right? So like maybe we could go into a cave and like expose the whole cave to strong UV light, or you could expose all the bats, but like, are you going to have the bats like hold their wings out? Like, what if it's like behind their ear? You can imagine that the logistics of that are also complicated. There is a lot of promise for vaccines. I do not work on vaccines, but there are a couple of groups that do. This article was published by the Washington Post very recently, just in October. I highly recommend it. It's a really nice article about the current research on vaccines for P. destructans in bats. Also a little logistically complicated. How do you vaccinate these tiny little flying mammals? But they're making some incredible progress there. And then the last area is the one that we're working on and a number of other groups are working on, and this is the idea of biological control. So one example of biological control that maybe you're more familiar with is if you are a gardener and you have aphids that are in your garden and you want to find a more natural way instead of pesticides to get rid of those aphids, you can go to your garden center and buy a bag of like 1,000 ladybugs. and put those in your garden. And the ladybugs are going to eat aphids because that's their preferred food source. This is a kind of biological control. So we are applying this concept to microbes in the places where bats live. So we are looking for bacteria and fungi that can fight P. destructans. And I'm going to walk you through the process of how we do that and how we assess different biocontrols. So this is what it looks like when we do our field studies. So we go into the caves, we go into the caves at the end of hibernation, so we're not waking them up too prematurely. We swab bats, we swab the hibernacula, all the different parts of the caves, we collect soils, and sometimes it wakes them up and they don't really like to be swabbed. And then we culture them. And so we're looking for all kinds of microbes. So we want to have as many diverse different kinds of bacteria and fungi as we can find. So we grow them on all different kinds of media in the lab. We isolate them, purify them, and then we test them. And the way that we test them is super simple. I'm 100% sure that every single person in this room could run this assay with like 20 minutes of training. So the way that we do it is we have a Petri dish with some agar in it. This is like food for our strains. And then we make a little spot of our potential biocontrol in the middle and we let it grow. And then we pour a layer of media that P. destructans likes to grow on it, because these aren't necessarily the same. And then we take spores of P. destructans, the pathogen, and we spread that over the top of this upper plate. If we have something that produces an antifungal compound, what we see is called a zone of inhibition. And so if this strain wasn't here, or if it didn't produce an antifungal, this whole plate would be just a fluffy white fungus. That's what PD looks like on a plate. But because this little culture underneath here produces an antifungal, you can see this zone of inhibition. And so we've used this assay to screen hundreds and hundreds of bacteria and fungi. This one is for bacteria specifically. We do a similar one for fungi as well. And a fairly large percentage of our collection produces some kind of antifungal. This assay is exceedingly slow. So I mentioned that they're psychrophiles. You have to grow them at low temperature. They grow very slowly at low temperature. And so unlike our human pathogens, where this assay takes 1 day, this assay takes 3 to 5 weeks just for one result. And so we try to just always have these assays running so that we can get through hundreds and hundreds of screenings. And then the really fun part comes where we do the chemistry. So if we have something that produces an antifungal, we want to understand what does it make and why does it make it and how does it make it? Is it a new compound? Is it a known compound? And so we use this process called bioassay guided fractionation, where we ferment our microbe, we extract it with different solvents, so things like ethanol and other solvents. We test them using different kinds of assays. We purify them using chromatography, and then we repeat that many, many times. And eventually, here's the other chemistry slide. It's going to be short. So for one of our strains, our most active strain, these are all the compounds that we purified out of that fungus. So we purified 18 different compounds. There probably are 50 in there, but I had to tell Yudi, the chemist, to just like stop. Like after 18, I was like, that's enough, like stop. When we purify each of these compounds, we can then test them individually, because we don't know if they're all antifungal. Are they antifungal in combination? Is only one of them antifungal? And so we can test them using a similar assay to the overlay assay that I showed you. The only ones that are active have this red asterisk. So it's compound number 1, compound number 11. That's it. All the other compounds are not antifungal in this assay against this pathogen. We don't know why they make these other compounds yet. But it's interesting for us as a chemist to have this whole collection of things that are very close to each other, because we can look at this one active version and we can say, this is the active one. Well, why isn't this one active? Or why isn't this one active? And that tells us something about the relationship between the structure and their activity. So the next question that we have is, okay, we have two compounds made by this fungus that are antifungal, but are they toxic? That's a big question. It's a big problem we run into. We find lots of things that are inhibitory towards pathogens, but they're often toxic towards healthy cells. And so the counter screen that we do is we test them against bat cells that we grow in culture. So we have different cell lines from bat skin that we can just grow in a little dish. We can put our compounds in there and we can measure how toxic they are, how cytotoxic they are. And so our two compounds, 1 and 11, they're not very toxic towards bat skin cells. So that's useful for us to know. So at first, that was sort of where I thought we would go with this project. Like we'd find some new antifungals. They wouldn't be toxic to the bats. We could put them out into a cave. And we were totally wrong about that. Because there are so many other questions that we have to ask and answer first. And so some of the first questions are shown here. Does it kill other friendly microbes? So I showed you some of those pictures of the cave and you saw that there were all kinds of fungi and bacteria and all kinds of things. You could even see them with your eyes. There's that much microbial life. We don't want to kill this pathogen at the expense of completely annihilating the entire microbial community of these very delicate environments. So this is a hard question to answer, right? Does it kill the other microbes that are native to that environment? We also need to know if our biocontrol can actually grow on a rock. Up to this point, we're just growing it on delicious media in a petri dish. That is very different than a rock. And then this question is also important. How and where and when would we apply our biocontrol in a given cave? These are not questions I was trained to answer in my PhD or my postdoc. So the first question, do biocontrol candidates indiscriminately inhibit everything else? And it might be hard to see where you are, but just to show you what this assay looks like, this little circle down here is P. destructans. That's the pathogen. This big circle on the outside is another friendly fungus that's very common in this cave. And so we can grow them side by side and we can see that this biocontrol doesn't inhibit this native species. And here's an example of some of those native species. Like there's lots of old timbers and wood and delicious nutrient sources in this mine, and we wouldn't want to kill all of this native microbial life. This is a data slide. I don't want you to look at the numbers. I just want you to look at the colors. So this was an experiment where we took the 4 most common fungi found throughout all levels of the Sudan iron mine. There's 4 of them. And we did a side-by-side assay between the 4 most common friendly fungi and all of our biocontrols. And we tried to see which one outcompeted the other. And the ones where our biocontrol did not outcompete these native fungi, we color green. And we give them a lower number score. So if you look down a column, the strains that show mostly green are the most promising, highly specific strains. They kill PD, but they don't kill these 4 common fungi. We can't test all the fungi. There's probably, I don't know, 1 million fungi. So we're testing 4. And we're currently in the process of doing this with bacteria. So this just gives us a sense of its specificity. How specific is this biocontrol towards P. destructans? Now getting to the question of does it grow on a rock? That's actually a really complicated question because the rocks in these hibernacula are all really different from each other. So I've been talking about the Sudan iron mine up here. So mostly that's jasper and quartz and iron. But in other parts of the state, we have sandstone caves in the banks of the Mississippi River. And in the southern part of the state, we have these calcium karst caves. So these are more like traditional caves that you might see stalactites and stalagmites. These are all important hibernacula for the same species of bats. So when we want to ask, well, does our biocontrol grow in that environment? There are so many environments. So I'll just show you how we're trying to address this question. So first we take rocks from a given environment and we boil them to sterilize them because we want to get rid of all possible things that can grow. Again, this is not something I ever envisioned I'd be doing, boiling rocks in the lab. I then made friends with a geologist and I got the keys to their cutting lab. And so now we can cut our rocks and sterilize them. And then we have these nice little, we call them coupons. So these are little slabs of rock from a given location, and we can do experiments on them with our biocontrol and our pathogen. But before we do that experiment, we need to know if either of these things can grow. And we also need to know what the natural microbes might be if they were just in the environment. So we sterilize them, but we wanted to see what they would look like if they weren't sterile. So this is what it looks like under a scanning electron microscope after we boil them. They kind of look like the surface of Mars. You don't see any filaments or cells. It looks like sand and rocks. Nothing is growing there. So then we took these sterile little coupons of rocks and we put them back into a cave for 2 months. So not super long. And we put them into an environment that was pretty dry. So no water is running through here. This is a cave that has lots of teenagers that come into it. So we tried to hide them so that like beer wouldn't get spilled on them. And this is kind of what they look like, right? They're just sitting out in the environment. We just didn't know what would happen. So it was interesting when we collected them 2 months later and we saw under the scanning electron microscope that they're covered in life. So all kinds of both bacteria and fungi have completely colonized the surfaces of these rocks. This version of these rocks might be a better substrate for the experiments that we want to do, because this is what it looks like in the environment. This is so complicated. This is so difficult. So this was interesting and also disheartening at the same time. We also want to know if we can grow P. destructans on rocks in the lab. We know it exists in all these caves. If you go into any cave that's infected and you swab a surface, you will be able to isolate P. destructans. We know it is everywhere, but we don't know if it's growing. We just know that it's there. And so we did an experiment where we tried to inoculate rocks with P. destructans spores, and we left them and left them and left them and left them, and they just never grew. So these are the spores of P. destructans. They look like little inflated commas. They're very distinct. And after many, many months of sitting on these rocks, they just don't grow. And so this is a real challenge because we had decided that we were going to do these experiments on rocks. But if our pathogen doesn't grow on the rock, then that experiment is not possible. So this is currently a work in progress. But I'll show you what we'd like to do. So when we talk about biological control and putting something in the environment, what we want to replicate is microbial life that already exists there. So these are pictures of, this is the Sudan iron mine, this is Mystery Cave, this is a scanning electron microscope picture of one of these colonies in the Sudan iron mine. These are all microbes that are growing. I took these with my cell phone. So these are not highly magnified, like you can just see them with your eyes. So there's all kinds of microbes that are very heavily colonizing parts of these hibernacula. And this is what we would want to replicate with our inhibitors, our biocontrols. Okay, so the other question that we want to know is, well, let's say we solve all of these problems. Maybe I should move less. Let's say we solve all these culture problems and we want to test them in a cave. We want to know where in a cave. Am I doing something bad? We want to know where in a cave we should apply our biocontrol, because these caves are actually pretty big. They're pretty extensive. We have this question: if bats become infected and they all die, let's say they all die and the cave is empty for 5 years and a new colony of bats tries to live there, will they become infected? We don't know the answer to that, but it's important, right? And then where would we treat P. destructans in that cave? because we can't treat the whole cave. And so this is a map of one of the caves that we study. This is Mystery Cave. This is the karst cave. This is the one that has like stalagmites and stalactites in the southern part of Minnesota. This scale is 250 meters. There are tens of kilometers of passages in this cave system that are known. And there's many more that have not been mapped. So it's a very extensive system. So this sort of illustrates why and how we could not treat a whole cave, either with a fumigant or with something that we would put on the walls. We have to be very strategic in where we would treat something. And so we did an experiment where we wanted to see exactly where PD was in part of this cave. So we did a transect. This is the entrance of the cave at this little star where this arrow is pointing. This is kind of what the inside of this cave looks like. It's pretty big. And this is the transect area that we focused on. And we looked at 9 different locations. And these locations are indicated by a green circle. And at each of these locations, we sampled from the ceiling, the walls, and the floor 3 times, combined all those samples, and then we used a technique called quantitative PCR to see how much pathogen was in that sample. And we really just wanted to see from this side to this side where PD was. We just wanted to make a map. And what we found is that the parts that are the deepest inside the cave had the lowest amounts of P. destructans. And then as we got closer and closer to the entrance at the star, we got more and more P. destructans. So that gives us some pretty useful information that most of the pathogen is at the entrance. So maybe we wouldn't have to treat all of this. When bats become infected with PD, they also tend to migrate towards the entrance. So this is where the bats are really focused. This is where they're hibernating anyway. So this location at the entrance had at least 10 times more DNA from the pathogen than location 7 and 8. But then Liam, the student that did this work, decided to sample the one door that keeps it's between the cave and the outside of the cave. And so the state park built this structure so that people can't just go in this cave. And they made the door with a hole so that the bats could go in and out before and after hibernation. And so Liam decided to sample the sill of this door where all the bats go in and out. And he found that had a lot more DNA. So clearly the bats are shedding P. destructans into the environment. And this is kind of a cool experimental setup for us because we can access this, we can clean it, we can measure it, we could clean it again, we can measure it. And ideally we would count how many bats are flying over it. That's a hard question to answer as well. Again, not something I was trained to do in my graduate training. So we acquired some bat counters, and these are big frames about this big. They have infrared beams, 16 on each side. So 16 here and 16 here. And if a bat flies through, it breaks the beam. It's called beam break technology. And it's just count it. And so you can count how many things break the beam. And so we can count how many things go in and how many things go out. And then about twice a year, we go and we completely disinfect that sill of that door. And then we come back 6 months later, we sample it, and then we disinfect it again. So we're trying to quantify how much PD is being shed by the bats. We still don't know how many bats are getting through here because we went to collect our data. There's like a little SD card inside, and we trudged out there. We had to bring our spare batteries. The batteries are like giant RV batteries, and we had them on a sled because they're so heavy and there's snow because it's Minnesota. And we had to like trudge out there with these heavy batteries. And then we noticed that none of the lights were on and that rodents had chewed through all the wires. I know, that's why they call it research. Okay. So you can see that we found more questions than answers, and every answer leads us to more questions, but we're learning a lot. We haven't solved this problem. We haven't cured this disease, but we have some promising data. So we're still working on it. So here's a summary of what we've done so far. We've identified at least 8 fungi that seem to be promising biocontrols. And among these, 5 of them do not kill the other friendly native fungi. We have also found that growing both the pathogen and the biocontrol strains on natural rock substrates is complicated, and we're still working on this. And we also know that P. destructans is concentrated near the entrances of the hibernacula, which gives us a target for where we would try to treat these locations. Some of our other challenges are field testing. So when we decide that we have the perfect biocontrol, we know it grows on rocks, we know it doesn't kill everything else, we're going to have to test this in a field location. And that's super tricky. Getting someone to give us permission to put a untested treatment out into the wild that grows, that you can't take back. So that's challenging. And then these bats that are dying from this disease are also starting to move onto the endangered and threatened species list, which also limits our ability to interact with them. Before I show you my acknowledgement slide, I just wanted to share this slide, which is a collaboration with a company called Minute Earth. If you are interested in learning more about natural products and their role in drug discovery, they contacted me to do a little video, a little cartoon on a complex idea. And they have lots of really interesting videos. I think it's 3 minutes long. And it tells you almost everything you need to know about the origin of natural products in finding new antibiotics, new antifungals, and anti-cancer agents. And this was a really fun collaboration. And with that, I'm going to show you my acknowledgements. So I'd like to acknowledge the funding agencies that supported this work. I want to acknowledge the students and the scientists that did these experiments. This work is done through collaboration with state agencies, with the Department of Natural Resources, and with lots of other groups. And so I'm super grateful to everyone who's worked on this project, and I thank you for your time and attention.